and welcome to That's Absurd. Please elaborate. I am Trace Dominguez, one of the science communicators who is sitting here in this call and will be talking about the sciences with the other person who's sitting here.
I'm ready. Let me go. I'm ready to jump in. Put me in, coach. I'm ready to introduce myself. It's me, the other one, Trace Dominguez. No! Dang it! That's... I was so close. Julian Huguet, that's me. I'm the other one. The other science communicator for this science communicating podcast. Is that our crutch word?
I feel like it is.
We also attempt to do comedy.
Once in a while. I don't know if it's comedy. I don't think I would call it that far. What's 3 steps below comedy?
Dad jokes.
Dad jokes. Yeah, I think that's more accurate.
Yeah. Your science communication / dad joke podcast where we take ridiculous, silly, outlandish, absurd questions from you, our listeners, and then we actually try and find a real answer, no matter how odd, weird, wacky the question may be to start out.
Yeah, that's true. I want to apologize a little bit. I apparently have a cold, so Julian's voice sounds buttery smooth, and mine sounds a little raspy, and I apologize for that. But I'll give my voice a rest and let Julian go first this week. So, by the way, if you haven't listened to this show before, thank you for listening to it. If you have listened to it before, welcome back. If you want to ask a question that we can feature here on the show, make sure you go down into the show notes and send it to us. You can call us on the phone, you can email us, you can go to our website. You can also join our Patreon and vote on questions that people have submitted for future episodes.
I'm so jazzed about the one that they just selected, by the way. I don't want to spoil it, but I'm really psyched about it.
I mean, by the time this airs, will, any patron at least will already definitely know that it's been selected.
But for listeners not on the Patreon, I don't want to spoil what's coming, but I'm very excited. I'm going to have to get in touch with people who do dog sledding. Anyway.
It's going to be so good.
That's the teaser. Anyway.
Oh my God. Okay.
Now, my question, though, for this week's episode actually comes from Annie Mouse. No, Anonymous. Excuse me.
Oh, Annie Mouse, our favorite submitter.
That would be a really cute little name.
Not actually our favorite. I prefer when people let us play their voices, but it's nice that people submit them, even if they're Annie Mouse.
Annie Mouse. So Annie Mouse asks, what part of the human body is the most heat tolerant?
Is Annie Mouse some kind of torturer?
I was going to say, the fact that it is anonymous and it's also like, what part of the body?
Excuse me. Hello, I have a question. for the podcast.
Yes.
If I were to drive a nail through a part of the human body, what part would be the most painful? I'm asking just, no, for reasons, you don't need another reason.
For science, of course. Well, yeah, I was going to point out, I'm very glad that it wasn't, the converse side of that, of like, which part of the body is the least heat tolerant? Where can I do the most pain with the application of heat? No, so hopefully, we're not aiding and abetting anybody nefarious.
Let's hope not.
Annie Mouse elaborates, I heated some pie.
Oh.
They typed air dryer. I'm going to assume they meant air fryer.
I mean, maybe they have no idea how heat works, and they literally did put their pie in an air dryer, and were like, "This isn't cooking."
or held up a hair dryer, and they're like, a blow dryer. Come on, pie. Anyway, Annie says, they couldn't lift it out with my hand because it was so hot, but I could eat it just fine. So I originally wanted to ask about hands versus mouth specifically, but started to doubt myself and decided to ask this more general question instead. And then, to make it clear that they are not, again, a torturous, murderous individual, they punctuated it with a smiley face. So we know it's fine.
Yeah, so it's fine. It's all definitely all fine. You put a smiley face at the end, it's all fine. That's the power of emoji.
Hey, get in my windowless van, winky face. Like, nothing bad happening over there.
Do you have candy? No.
Well, I don't get into windowless vans for anything less than full bars. Not even fun-size Snickers. No way.
Ugh. And what is the... Okay, we're recording this not long after Halloween. I do have a kid. What's the size smaller than fun size? Because when I was a kid, fun size was like oh man. And now they have like the tiny cubes.
Yeah.
That are like 1 centimeter of candy, 1 cubic centimeter of Milky Way.
Oh, okay. Well, on the fun sizing question, to take a totally unrelated tangent.
Yeah.
Is it like the smaller you get, the more fun it gets?
Like, that's the implication.
I have more fun when I get a larger bar. Like, I don't look at a full-size bar and go like, wow, that's serious. I'm like, this is better, right?
This is going to be great.
It's always like king-sized.
Yeah.
I guess fun size is like jester-sized. So then maybe it's a royal court.
And so that would be more just like lord-sized, just the little cubic centimeter. Surf-sized?
Surf-sized. Surf-sized.
Surf-sized.
SERF. That's the portion Aaron Aronson gets when he goes trick-or-treating.
Oi! I would like a larger Mars bar, please.
Oi!
This is too small. It is just 1 cubic centimeter of chocolate.
Please.
Please more. Please,
My boy has the plague. Let him taste something delicious before he goes. Also, what's chocolate? Because he's from before the Europeans learned about, the new world, quote unquote. So yeah, very surprising. Any size chocolate back then would be fit for a king.
To be honest, true. Yeah.
Yeah. That all had nothing to do with anything we're about to talk about. So Here we go.
Again, if this is your first time, thank you for listening to this podcast.
That was a test. You passed.
Now that all of the new people have left.
now we've weeded out the normies. Okay. Anyway.
We're not great at podcasting.
No, we're going to have to stop using that science communicator word. Okay. This made me think of that tweet, though. I think it went viral where somebody asked, like, do you blow on your food to cool it off, or do you just that until you can eat it?
Oh.
Which I felt very relatable to me. But I started thinking first off about, any part of the body that's most heat tolerant. And my mind immediately went, of course, to your bones, right? And it went to specifically cremation.
Oh.
Because I'm a bit morbid.
Yeah.
And I also watched that ape crazy or chimp crazy docuseries about how they tried to cover up the death of a chimp. Like, they faked the death of a chimp, and then they had some ashes. It's a crazy documentary. I won't get into it. But anyway, some people who illegally had a chimp tried to say that it died and was cremated.
What?
And I was watching it there's no way. But anyway.
What?
Watching that led me to look up some things about cremation. Do bones not turn to ash when they get cremated? And it turns out, a lot of bone fragments stay behind. So the cremation ovens are 1,400 to 1,800 degrees Fahrenheit, or about 800 to 1,000 degrees Celsius. And at those temperatures, the soft tissues turn to ash like you would expect. The bones, though, calcify. They don't just fall apart. They might break up into smaller pieces, but you'll still get these little bone fragments. So, hot tip for all you murderers out there, you still have to grind up the bones. At a crematorium, they have a machine called a cremulator, which they use to grind the large pieces of bone down. So it's like a sand-like consistency. And then, you can have your little urn of your beloved, consistently ashy, sandy, smaller, fragmented former loved one. And rest in peace knowing that they're all smoothed out.
So sentimental when you put it that way.
I know. It's weird. I hope, if they cremate me, I hope they leave the bone fragments behind. So if somebody tips over the jar, you have to pick up some larger pieces. Anyway.
You just have to vacuum you off the floor. Yeah. And then the vacuum becomes the urn.
The vacuum is the urn?
And then you just hang the vacuum on the wall and people are like, oh, you spilled something. Let me go get your vacuum. And you're like, no. That's Julian. You can't. And they're like, Julian, I've never heard of that brand of vacuum. I think it's a Dyson.
Yeah. Knowing me, it'll be a dirt devil. Okay. So then I realized that that's not really what Annie Mouse is asking, right? They're more talking about sensation when you're touching things. What is the most heat tolerant, right? Because when you have hot food and it's burning your hands, but then you just shove it in your mouth, and you it may feel more tolerable, but it can still burn your tissues, right? You can burn your palate, you can get blisters on your cheek, on your tongue. So it's still doing damage, but for whatever reason, you're perceiving it like it's more tolerable.
Yeah.
So I did some digging onto what could be possible explanations. My first thinking was about just how sensitive our hands are in particular. right? Like when we talk about nerve endings and where they're concentrated in our body, these different nerve endings that help us feel sensations, there's a ton in our hands, and particularly in our fingertips, which makes sense, right? We use them a lot to perceive the world around us. So I thought that might be some possible indicator of it. But of course, there are a lot of different types of nerve endings. So the kind that we're interested in, of course, are those that can feel temperature. And there are many. And so I looked into that, and there are specific nerve endings that have to do with different temperature ranges. For example, there's a receptor called TRPV1. It stands for Transient Receptor Potential Cation Channel Subfamily V, for vanilloids, number 1.
Wow.
And it's sensitive to temperature, which is thermoception, but it's also sensitive to pain, nociception, right? And the way this protein works, basically, it's a complex protein that's like in the cell membrane of your skin cells, and it gets deformed in high heat. And when it does that, it sends a signal through your nerves to your brain, and you sense that heat. But it also can be triggered by molecules that are released when other skin cells get damaged, say something like hits your skin, pierces your skin, it destroys some cells. These cells, as they burst, release everything inside of them.
Yeah.
And some of these trigger the TRPV1 receptors, and that sends a signal to your brain as well. And so you interpret that as pain.
That makes total sense, though, because you would want to know if your cells are being bursted.
Yes. Damaged, or if the heat of something is enough to damage your cells and you need to quickly recoil your hand from the hot stove or whatever, right? So it's not only the temperature sensing, but also the, that hurts sensation. Yeah. Another thing that can trigger it, I mentioned that the V in TRPV1 is for vanilloids. You know what one particular very famous vanilloid is?
Capsaicin.
Yes!
I have a whole talk about this.
Good.
And it's why we have spicy butt.
Yes, that was not the particular phrase I was going to use, but capsaicin is the molecule that you find in like chili peppers, and it triggers this TRPV1 receptor, and that's why spicy hot and temperature hot can be kind of conflated, right? You know the chili pepper isn't over 43 degrees Celsius, which is how hot it has to be to trigger these TRPV1 receptors, but still causes that burning sensation.
And your body doesn't know.
You're just working, with the best input that you can get from outside stimuli. Like, it's, sometimes things get conflated. You also have receptors that are sensitive to cold, like TRPM8, and TRPM8, along with low temperatures triggering the nerve response, also can be triggered by menthol. So that's why you get that cooling sensation.
Yeah.
Very cool.
Cool butt.
So...
I don't think people eat enough menthol to get that.
The only cure to spicy butt is... Trace, do you want to explain what you're talking about when you say spicy butt in particular?
Fire poops.
Right. You gave a talk on this, apparently?
Yeah. I did a nerd night on it.
Oh.
Yeah, because I wondered why our butt could taste spicy food. Which really is a question for this podcast, to be honest.
It is, but we've already answered, apparently. So, I thought maybe, the fingers have more TRPV1 receptors or something like that, but no, they're also jam-packed in our mouths as well and on our tongues. Our tongues also have a lot of nerve endings, which makes sense because they feel the food that we're eating, the texture, and obviously the flavors based on the molecules present in the food. So I don't think it can be put down alone to saying, well, our hands have just more of these nerve endings and therefore they're more sensitive to heat. But I couldn't really find any research that went into it as to why, right? Why when you shove something hot in your mouth, it seems more bearable. I do have some potential explanations that I found or that I thought of that I think are reasonable, but I couldn't find any dedicated studies to it. I think, first of all, you've got your mouth is at a higher temperature than your hands, right?
Yeah.
It's inside your face with the mouth closed. It gets warm from the biological processes, so it heats up to body temperature.
Closer to your basal body temperature.
Yes, whereas your fingers are touching the air that's, around room temperature, most likely. So maybe because there's less of a temperature difference between your hot food and your mouth than your hot food and your fingers, that's one reason that maybe it doesn't seem as bad. You have saliva in your mouth, which can absorb some of the heat of your food, right? As it soaks up that energy from your food because a lot of it's water. You've got airflow from breathing, right? So you're kind of like fanning it as you go, right?
Yes.
And some of that heat is being absorbed by the air that you're passing over it and being carried away. You're also moving the food around in your mouth so it doesn't sit in one place for as long, right? If you've got a hot potato, let's say, and you juggle it from hand to hand, you can withstand it longer than if you're just holding it until it burns your fingers.
Right.
And of course, when you take a bite of something, you're just taking a little bit of that hot thing. So it's easier for your mouth to cool it down. It doesn't have to absorb all the energy in the whole thing of food than when you're holding, I guess, your entire pie out of your air fryer.
Air dryer.
Air dryer, right. I did find a 2007 study from the Journal of Sensory Studies on why coffee doesn't burn the mouth. They said that they actually monitored the coffee temperature with thermocouples in the cup, in the coffee bolus, in the oral cavity, on the surface of the tongue. And they found that there was minimal cooling as the coffee went into the mouth. And the coffee temperature was still above the threshold for inducing burn damage. So they hypothesized that during drinking, the bolus of hot coffee just isn't in your mouth long enough for it to heat the epithelial surfaces and cause pain and tissue damage. That was it. So basically, because you're just slamming back the coffee, you're like, it burns, but not very long, so it's fine.
That explains why then you burn your mouth with pizza. Because that stuff's in there for a minute, and it's like, cheese holds a lot of heat.
Yes, as we learned from our moon cheese episode.
Yeah.
Right, it can get quite hot. So I hope that answers the original question that Annie Mouse thought of why can I shove it in my mouth and it seems okay? But I wanted to also look, definitively, can I find any research on the most heat-tolerant part of the human body? And Trace, would you believe it? I found a paper on this.
I mean, I would believe it. There's a lot of papers out there, but is it a good paper?
Now, that's the thing. Reading it in my unexpert reading, I don't know how convinced I was in their findings, but I will get into that. So this paper was from January of 2020. It was from researchers at Cal Berkeley, and they wanted to map out the thermal sensitivity of the entire human body.
Oh, wow.
And this is because they are working on, quote, personal comfort systems. Basically, just like some clothing that you can wear that if you're cold, it'll heat just you up. And the idea is this way you don't have, like in a big office building, to set the temperature to something higher that would use a lot of energy or colder, whatever the case may be. You just wear some personal clothing. And if you're like, I'm feeling a little chilly, you just dial it up on yourself. And then people in the office don't have to fight over the thermostat, that sort of thing.
Yeah.
So it saves energy, hopefully, makes everybody get along in an office workspace. But in order to know, all right, how should we design these to be most effective, they wanted to actually measure the different heat tolerances across a person's body. So they had over 60 people come in, men and women, college age. They controlled for like different sizes typically. So they selected like larger than average women, smaller than average men. And they probed them with little...
Whoa.
Sorry, that sounds more intense than it was.
Wow.
They just stuck a little thermal probe on different parts of their body. They tested 318 spots. They would either warm this probe up or cool it down, like plus or minus 5°C. And then they asked the participants to rank how hot they perceived this change to be. Was it really hot? Is it not noticeably hot? What was it, right? And I have included, I just sent it to you, the map that they made of the sensitive regions of your body to different temperature changes.
Whoa, what a booty.
Yeah. Well, specifically, Trace, what do you notice about the booty?
In my mind, when I first saw it, I was like, there's a shadow underneath the buttock, which makes sense because that is a badonkey one. But that would be a very sensitive temperature area.
Yes.
Both for cooling and for warming. How interesting.
According to this study and the data they got, the most sensitive part of the body is basically right under the buttock.
Yeah, like the butt crease.
And I don't know why that would be the case.
Yeah, that is very strange.
Just kind of the area if you're having like a really intense makeout sesh, like where someone would grab you, you know?
Yeah.
Does that help elucidate the region I'm talking about, listener? I sure hope so.
I hope so.
Anyway, now, about this paper, about this study. Again, they worked with over 60 people, but they broke them up into 3 groups, I think likely, so they didn't take up all their time, like going over all 318 spots on every person, you know? So It was really more like 15 people per sex, per group, thereabouts, that they probed, which I don't think is a huge sample size. And the fact that the scale that they used to rank the sensitivity was very subjective also makes me wonder if that might confound this. I have a theory that because they were probing under the buttock, which is quite a private area, that unless, you know, you're doing a hardcore makeout sesh with somebody that normally I don't want anybody to touch, I feel like probing me there would make me more acutely aware.
Agreed. That's exactly what I thought. I agree with you that I'm skeptical. I don't think it's actually that area is significantly more or less sensitive to, or like more sensitive to hot or cold. It's just that area of the body, that like under butt, isn't exactly typically stimulated in many ways, right? Like You're not sitting on a chair and that part of your body is all of a sudden twinged. That is a part of the body that is, as you say, more intimate. And so that's when it is touched. That does also, just to counteract my own argument, maybe that's why it is more potentially temperature sensitive, because it doesn't get as much interaction directly.
Yeah. Well, speak for yourself. I don't know about you. Well, I almost wonder, though, because, it's like a researcher probing them with this temperature probe, if that made it more awkward. But if they were to sit on like a heated seat or something and get warmed up the same way, would they rank it as less sensitive? I don't know. But fortunately for us, that's not really what Annie Mouse was asking about. It was what's least sensitive. And that, I think, is more believable based on these graphs that we have. And it is like the shoulder area, kind of the top middle of the chest, and then what I suspected from the get-go, which would be the feet, the extremities.
The feet, yeah.
Bottom of the feet. That makes a lot of sense to me, right? That is a part of your body with some thick skin. It's got to walk over rough terrain or like hot or cold, right? It's probably more suited for touching high temperature stuff longer than the rest of your body. So that, to me, I think would have to sort of be the answer. Things that you can tolerate physically, right? Not like if you put me in a cremator and what's left, my bones. Things that I can touch onto a hot thing for an extended period of time and still feel pretty okay with it, probably going to go with the bottom of the feet. Now, in a slightly related note, though, right? If you are having a hardcore makeout sesh, before you get to your air-fried pie with your significant other, right?
Your air-dried pie.
Your air-dried pie. And you want to particularly make things real hot, we now have a published, peer-reviewed chart of the place that gets hottest when you're stimulating, and that is under the buttock. So
under the buttock. However, if you don't want to go all the way to the to third base, I think it's called, you could go midway, and around the ribby area seems to be kind of nice.
Yes, we'll publish the chart on our Instagram. So if you're using hot or cold jellies for whatever reason. Anyway, well, Trace, now you can add a corollary to your spicy butt talk.
I can.
Like, hey, not only can your butt taste spicy, but also it feels the heat probe the most.
Look, I didn't realize I was going to get more fodder for my conflagulatory buttock talk, but here I am, learning things all the time.
Yes, this is what science is. We're building a foundation of knowledge, right?
Correct. We're starting at the bottom.
That's great. Well, Annie Mouse Anonymous, I hope that answered your question and you can get back to cremating that body or whatever it is that you're doing.
We're not asking. We're not asking
the questions here. What are we, cops? Not us.
Yeah, why don't we come back in a minute with my question. Julian, give me your best dinosaur roar.
Oh, God, an asteroid! It's horrible!
Wow, that was not where I expected that to go. We're popping in before the show to tell you about I Know Dino. It's a dinosaur podcast.
You know, we think of those dinosaurs being this thing that's been dead for tens of millions of years. Even so, studying dinosaurs can still teach us stuff that is relevant to the world today.
Yeah, like climate models to help us understand global warming.
Migration patterns of dinosaur lineages, they tell us about Earth's changing continents.
We can also study dinosaur diets, what plants lived then, and maybe what animals and how they interact.
And, this is super cool, many dinosaur injuries, paleopathologies, they're the first known occurrences of diseases.
And they talk about all this kind of stuff on I Know Dino. It comes out every week with new dinosaur stories that you're not going to hear anywhere else. Like, I hadn't heard of a lot of these, which is pretty cool.
You can find I Know Dino on Apple Podcasts, Spotify, wherever podcasts are. And if you found us, you can probably find them. Go check them out. Welcome back to That's Absurd. Please elaborate. We hope that you're toasty warm as you're listening here. And again, we're very northern hemispheroic in our perspective. We also hope you're staying cool if you're down in the southern hemisphere.
Yeah, I hope you're having a good summer.
If you're in the tropics, we hope it's just whatever temperature it always is, apparently.
Ugh, yeah, just brag.
Yeah, aren't you special? Trace, what was your question this week?
My question this week came from Maikomo or Mikekomo or I don't know. They didn't send their name, but they did send their email. So it's not their entire email, but I took a part of it and I said, it's their name now. So Mikekomo, if you stitched together all the DNA on Earth into one strand, how long would it be?
Ooh.
Now, on its surface, it seems like a very simple math question, but they did elaborate. I don't remember where this question came from. In my mind, I've convinced myself that it came one day out of nowhere, but it's just as likely that I read it in a book and took credit for it subconsciously. I do remember searching the internet at the time and not finding any references on it, but doing the same research now comes up with a few hits, so who knows. I had this thought. The human genome is something like 2 meters long. So how much DNA is there in total? I ended up doing this back of the envelope calculation and figured out that it would come up with a good fraction of the universe in length. I also think there's this really cool aspect that combines a really small thing with something really big in a way that our minds can't really grasp the scale of, which I love that. So anyway, they said, I was floored by this answer that it was a fraction of the universe, and I convinced myself that I must have done the math wrong. I'd like to hear your take on it. Mike Como.
I would like to ask somebody possibly less qualified by the sound of things to look into this for me.
Right. So I like these kind of questions. I've done several of them on the podcast, so this one really stuck out to me. It does feel like a simple math question, right? And actually, Mikikomo did a big part of the work for me in that, yes, DNA in a human is roughly 2 meters long, or about 6 feet. And so you could just multiply the amount of DNA by the number of humans. And I did that, but let me come back to it in a minute, because not everybody probably understands all of the story of DNA, which I think is really interesting.
Oh, okay. Are we going back to the beginning, like primordial soup time, or like...
No, just the 1860s.
Oh, good.
DNA was first spotted and identified by Swiss biochemist Friedrich Meichner. He called it nuclein. My friend from Austria says when E and I are walking, whoever walks first does the talking. And so I think it's nuclein. Anyway, he called it nuclein because he isolated it out of cellular nuclei. And he thought, wow, what a cool new novel molecule. It's really interesting. It's not a protein. It's not a lipid, like a fat. And it showed up in these other nuclei, too. It just seems to be in all these nuclei. And they figured it had something to do with how the nucleus worked and whatever that was and what it was doing. And actually, this guy, Friedrich, was like, maybe it has to do with heredity. Nah, the molecule's too simple.
It's so close. Too simple.
Yeah, 1869. Because they were like, oh, well, right before the cells split, there's a lot of nuclear substances inside the cell. So maybe it's heredity, but nah, the molecule's too simple, so it must be something else. So over the next 80 years, they start to figure out what DNA does. And finally, in the early 1950s, Rosalind Franklin, James Watson, and Francis Crick are working in a lab in the UK. Franklin was an expert in X-ray diffraction, allowing scientists to actually see molecules like this, other molecules too, DNA as well. But she was studying coal at a university in Paris, weirdly, before DNA, like the structure of coal. That was her job, which is also very British of her to be like, let's figure out how coal works rather than figuring out anything else in the world. We'll just keep using coal forever.
Let's see if we can make super coal.
There's not enough soot in London. Let's get some more coal in here. Let's figure it out.
Great Rosalind Franklin impression.
Yeah. No, I was being Rosalind.
You were being the coal baron financing Rosalind Franklin's research, yeah.
Yeah. So the scientists at this point already knew that DNA was phosphorus-based. It had this like phosphorus backbone with these 4 different base pairs, but they didn't necessarily know how they paired together. They knew that they were there. They knew that they had adenine and thymine and cytosine and guanine, but they didn't know that A always went with T and C always went with G. They did know what the molecule was. They knew it was there, and they were trying to figure out more about it. And so Franklin differentiated with X-rays 2 forms, A and B. She was the first to do that. No one knew that it was 2 forms of DNA, and she did it by raising and lowering the humidity where the DNA was being held. And at high humidity, you get B form. Low humidity, you get A form, which is super cool.
Very cool.
A form was easier to image, so she focused a lot on A form because when it was A form, I assume it must have laid flatter or something. Like it was just something about it was easier. The image was sharper. In her notes, she wrote, it was a, quote, big helix with several chains, which is cool. She didn't really like the B form because the pictures ended up much blurrier. She just couldn't get as good a picture of it. The A form, you could see more, which meant there was more to study. So it's actually more complex to understand it. But the B form, she couldn't get a clearer picture, but it would have been simpler to understand it. So she's focused a lot of her time on the A form, which I think is really interesting.
I wonder if that's kind of where the A form and B form comes from, because if my memory serves, when we talked about cloning a left-handed you, B form is the most common form that we actually, like, our cells use as the blueprints for us.
And we didn't know that at the time. And what they figured out later, what basically she had overlooked, and this isn't necessarily a critique of her. This wasn't, I'm not critiquing necessarily, but it's just that,
where's your Nobel Prize?
Right. That cells are wet. And so saying like, oh, let's study all of this A-form stuff in this dry environment, DNA is going to live inside of the nucleus, which is a wet environment. So B-form might be more common just because it's wet inside as well. So anyway, enough about the DNA. Let's talk about that hot sciencey goss.
We have to get into it if we're going to talk about Watson, Crick, and Franklin.
Right. So Watson's book, which came out much later after they'd won the Nobel Prize for, quote unquote, discovering the structure of DNA, he wrote that Franklin was preparing to transfer to another college while working with them, and they didn't get along, Watson and Franklin. And so they were all kind of working in silos, each of these scientists. Watson and Crick would work together, and Franklin would kind of work separately. She was instructed to leave her DNA research behind with Watson and Crick when she went to King's College, London. And so Franklin's grad assistant showed Watson this photo called Photograph 51, the most famous, probably, photograph in the biological sciences. It's the photograph of DNA. It is a blurry image that if you don't know what you're looking at, you'd be like, I don't understand what this is. It was taken 8 months before she left. She made notes on it. She studied it. But like I said, she didn't really care as much about the B form, and the A form were clearer. So it's not that she didn't care. She was just like, I can study more of the A form. I can see more of it. And her job was to study the structure. So Watson said in his book that he saw this blurry picture and immediately knew it was a double helix, which modern critiques of that narrative are like, no, you didn't. You don't study crystals. Like, you don't understand. You would not have, no one would have looked at this blurry photo and been like, my God, a double helix crystalline structure. Like, nope, nobody. Nobody would have done that. She was a chemist. She knew that it was a crystal. She knew how it worked. And he was not a chemist. He was a theorist. And so it's like most people have called BS on this. They say the reason that Watson and Crick in part are the famous ones and Franklin is not is because journalists and novelists wanted that eureka moment, right? They wanted that moment where he got this photograph, he saw it, and he knew it was a double helix. But here's the real story, which I thought was really cool. Nature has a write-up on it, really interesting, where they wrote, Crick's supervisor got an unpublished report from King's College London, where Franklin had gone to work. Now, scientists do typically share their findings, but the King's College people didn't know it had been shared. It was unpublished, and it was just like, oh, hey, look at this thing. That they're also working on the structure of DNA. That report included a page with Franklin's work on it, amongst other people. It was such an interesting report to them because it confirmed and like shored up some of their theories, because again, they're theorists. So after the King's report was published, it really pushed Watson and Crick to be like, let's try some of these theories out. They got some cardboard together, and for 6 weeks, they were just doing trial and error structures of what DNA might look like based on what they knew. And after a while, they figured it out. And they didn't actually cite the King's College paper at all in their research. They said they'd use their trial and error, and like that was the story they wanted to tell, and they didn't cite Franklin, and they didn't cite the other researchers. They said, oh, it was the environment of science at the time, which included this paper and Franklin and these other people.
Right.
But they were like, oh, well, they were talking about it, but we figured it out. And it was like, no. Franklin discovered that there was an A form and a B form. Previously, people had been taking pictures of it using X-ray diffraction, and they were like, a mix of A and B form, so it would always come out blurry. She discovered the DNA was C2 symmetrical, which is pretty cool, or at least theorized it. The main thing is Franklin as a woman couldn't have these like casual conversations in informal clubs and hangouts with male scientists because she wasn't invited, and she couldn't bounce ideas off of them because Watson and her didn't get along. And so Watson and Crick got a Nobel Prize, and Franklin didn't really even get recognition until decades later. But what we learned, and what they got a Nobel Prize for, is recognizing the double helix structure of DNA. So even though each human cell does contain 2 meters of DNA, which seems like a lot for every single cell, it's wound in a double helix into these little teeny packages of chromosomes, 23 chromosomes for each person, so 46 in a cell, of one from each parent. I don't think most people appreciate what a double helix really is, right? We picture that twisted ladder, and that's it. Mm-hmm. But there's actually a separate twist.
A twist to the twist?
There's a bigger, like if you zoomed out, it would also be twisted again into like that roller coaster style helix. And so it's a double helix, which means that you can twist it and then twist it up even more to make it extremely dense, which they, when they were taking pictures of it, even in the 1950s, were like, this is very dense. There's a lot of stuff in here. So that really was intriguing to them. So let's talk a bit about Mike Ikomo's question. Again, simple math. 6 feet per cell, 23 pairs of chromosomes from each parent. So you have 2 sets of instructions in each cell for making a full person. Your body uses some from each of those chromosomes. And you have about 6 billion base pairs total, or about 3.1 billion base pairs per 23 chromosomes. So if you do the math and you say the number of cells in a human body varies, depending on your estimates, between 20 and 40 trillion. So in every one of those tiny cells, there's 2 meters of DNA. So 30 trillion, which is right down the middle, times 2 meters equals 60 billion kilometers, which is a lot.
How many Golden Gate Bridges is that?
Well, it's funny that you ask. It's 150,000 times to the moon and back.
Whoa!
So that would be a lot of Golden Gate Bridges.
Oh, take that, Apollo space program.
Yeah, it's enough to wrap around the Earth 1.5 million times. And since to the moon and back is still too big a number to really grasp, it's 200 times to the sun and back, or 5 times to Pluto and back.
Wow!
It's 401 AU.
Distance between the Earth and the sun, yeah.
Right. And the reason it's 401 AU, I did 200 to the sun and back. So 400 AU trips.
400 single one-way trips. Yeah,
one-way trips, or 200 round trips. So if you think of there's a little over 8 billion people on our planet, that's just one person 400 AU is one person.
Wow, you have so much potential inside you. Every one of you.
So 400 AU times 8 billion people equals 3,208,000,000,000 astronomical units.
Okay, so how many Golden Gate Bridges is that?
I mean, I could do the math. Do you want me to do the math? I can do the math.
No, it'd still be a meaningless number.
So that's a DNA strand that's 50.7 million light years long.
Oh, wow.
Which is a string stretching from Earth, or let's just say the sun, because at that point, who cares?
Who cares?
You're talking a few hundred. Who cares about a few hundred astronomical units when you're talking? It's like, who cares?
8 more light minutes, like big deal. Okay.
Right, yeah. All the way to spiral galaxy NGC 4951, the Seyfert galaxy located inside the Virgo constellation, or the Needle galaxy NGC 4564. So maybe, 50 million light years away. That's big.
That's big, but I can't help but notice that's humanity's total genome length.
Great point, Julian. Somebody's listening.
I seem to recall the question was not how far could a human or all of humanity's DNA reach. It was all of it.
All of us.
All the DNA.
All the genomes. So the universe's diameter is 93 billion light years. Keep that in mind. Put a little pin in that.
Oh, yeah. I never forget that.
50 million light years away, which is all the humans, is about 1/1833rd of the universe.
Okay, it's not that far yet.
It's long, but we aren't that far out.
Yeah.
So to add every animal is really... Mike Como, this was mean because genomes are not all the same size.
Right.
Some things have bigger genomes than we do, and some things have smaller genomes.
We have fewer chromosomes than a potato.
Exactly.
If I remember correctly. But I don't know the base length of the potato chromosomes, right?
Right.
Like how many base pairs each chromosome is on average or whatever.
Do you know what the shortest genome that we know of right now is?
So when we talk about shortest genome, my mind goes, it's got to be a prokaryote, right? Like eukaryotes.
It's a bacteria.
Yeah. Eukaryotes have a nucleus. They have typically like a lot more, the cells are a lot bigger, there's a lot more DNA. Whereas prokaryotes like bacterium and archaea, they have shorter genomes. Bacteria even have like basically a big roller coaster loop, and that's like the whole genome. So I would think it's a bacterium.
It is a bacteria, Carcinella rudii. It's a symbiotic bacteria with 182 protein coding genes, or 160,000-ish base pairs.
Oh,
pretty short.
Yeah, that's short.
19,000 times shorter than yours.
Is it called Rudii after like Rudy, the diminutive Notre Dame football player? Because it's just a little guy.
I don't know, but he's just a little guy.
It's just a little guy, and it's his birthday.
His birthday. And
it's
honestly,
his birthday.
Both of those statements are 100% fact.
Okay.
Because there's been some Carcinella rudii born in this exact moment that we're speaking whenever you're listening to this. So it is.
It always is. It's always his birthday.
Always Carcinella rudii's birthday. And you know what? My headcanon is yes. The answer is yes. I don't know if it's true, listeners, but yes.
I'm imagining a little bacteria birthday party now with like the little hats. And then he's got like a pie that he puts in his little bacteria mouth and it's so hot. He's just got that.
They have a piñata and it's just like some kind of other bacteria that they hit.
Yeah, right. It's like E. coli and it bursts open and like spews.
And they're like, yay, cellular contents.
Yeah, totally.
So that's the shortest genome on the planet. The longest one we know? Any ideas there?
Okay, I think I actually looked up this fun fact a long time ago.
Oh, yeah?
It's like a flower that has some absurdly long, I want to say it has over 100 chromosomes or something like that, and it is known for taking like ages and ages to grow and reproduce. Am I right? Do I remember correctly?
It's Themyscteris oblanceolata, which is native to New Caledonia and Australia, and it's got 160.45 billion based pairs.
Wow.
That is a 300-foot long, 91-meter strand of DNA.
Wow.
50 times longer than yours.
That seems inefficient.
It's not a flower, though. It is a fern. and it's very small. It's only like
it's a fern.
It's only like 6 inches tall, 15 centimeters tall. It's just a tiny little fern, and it just lives near like water and just hangs out, and you would not even notice it. One day they sequenced it, and they're like, "There's a lot of DNA," and I made a mnemonic so you'll never forget it.
No.
Tio Blancy, Olata, it's got a big genome. Gosh, I wish that we knew why. You're never going to forget it now. You're welcome.
No, I have to say,
Tio
going
Blancy,
back.
Olata, it's got a big genome. Gosh, I wish that we knew why.
When I was doing my research on hot things in your mouth, somebody called it, it's triggered when you put like a lava pocket, like a hot pocket, but a lava. And I wanted to be like, no, it'd be a magma pocket because it's still under the surface of the bread. It's still under.
Oh, that was great. I'm so happy.
So bad at that.
So
okay.
Great. I love it. So
that makes some sort of sense to me because like ferns have really bizarre life cycles. If you ever look up, because they're ancient plants, they're like gymnosperms.
They are, yeah.
And if you look up the way that they grow between different generations, they look like totally different plants from one generation to the next.
Yeah. Ferns are fascinating because they're one of the like earliest plants, really. Like they're ancient plants. And so if we can understand why this plant has so much DNA, that might teach us something, but we really just don't know. There was a really great episode of Science Friday on NPR where they talked about this plant, and they said that it just has this really long genome, but there's an evolutionary pressure to have the right length of genome. So there must be a reason that it has this long genomic length. Because, we've done videos about this over the years, and they don't need all that DNA to make the plant. A lot of that DNA they call non-coding DNA or junk DNA. And I've been chastised for calling it junk DNA before. That was a common term for a while, but it's not actually junk. It's just non-coding. We don't know what it does.
Yeah, well, we don't need a ton of our own DNA for that matter.
Right.
Like, there's lots of nonsense DNA that doesn't get transcribed at any point and used.
And it is useful for things. There was a study, a June 2024 study from the University of Colorado that looked into non-coding DNA specifically to find out what some of it might do. And they think that there are regions that contain sequences, quote, that are known to affect gene activity, basically gene expression. And so there are a few popular and commonly discussed ones are enhancers and repressors. So it might say affect why some people are at risk for type 2 diabetes while others are not, or some types of heart disease. And they don't know. They're still learning what the non-coding DNA does. The thing is, though, the reason there's evolutionary pressure to have the right genome is because DNA is made of stuff. So you have to consume from your environment nitrogen, phosphorus, in order to build a new piece of DNA. And if you have a really long genome, 50 times longer than ours, that's a lot of resources for a little guy.
Just a little guy.
For just a little guy.
Just a little fern.
So if we have a average amount of DNA, I don't know if that's actually true, but like we have 2 meters, and then some things have teeny tiny genomes, and some things have really big genomes. How do you determine how long our genomic strand to the stars would be? Also, genomic strand to the stars would be a really
great band name. Oh, yeah, baby.
That's electronic. Oh. Definitely electronic, but like psychedelic electronic.
New age rock, like Fish. It's a long jam sesh, basically.
Just 1 5-hour jam sesh. Yeah.
Takes a long time to get to the stars.
It really does.
Gotta have some tunes.
If you're going to go, you may as well listen.
For the road trip, yeah.
To genomic strand to the stars. So how do we determine the distance of this DNA? All the organisms are different sizes. Do I make a spreadsheet of every organism? And I'm just like, okay, humans, 2 meters. The T-O blank C, O la ta.
No, stop.
91 meters.
Do you take some representative samples, like a bunch of organisms, and do some statistics and get like a median and then a standard deviation? I don't know.
So, luckily for me, I didn't have to, because a 2015 study in PLOS Biology guessed something even simpler, which was, rather than thinking of organisms with DNA in them, they went to just number of base pairs on Earth.
Okay.
Total number of base pairs. They just said, hey, I don't care how many DNA base pairs you have, or a virus has, or a plant has. What's the total number of base pairs on the planet? It was written up in the New York Times by Rachel Newer, and she wrote, quote, Earth is sort of a supercomputer in which total DNA would represent the system's storage capacity and rate of genetic transcription and its computing power. I don't love that comparison, but it is data storage and retrieval, so that's pretty interesting. And they thought, okay, they estimate if you look at the biomass on the planet of plants, animals, microbe, fungi, viruses, together, all of that would weigh 50 billion tons. Just the DNA.
Whoa.
Yeah, that's 45.3 billion metric tons of DNA. If you want to visualize that,
how many Golden Gate Bridges is that?
They describe it as 1 billion shipping containers.
Wow.
That's enough to go to the moon and back 15 times, 300 times around the Earth. It's about 12,000 kilometers because each shipping container, a standard shipping container, is 40 feet long, or about 12.2 meters. So because of that, we have some math we can do. Because they didn't just give us the mass, like the actual biomass. They also said the Earth's number of base pairs. Any guesses based on what I've just told you? And be prepared to be way off because it's a ridiculous number.
I'd have to guess like 10 to some power.
Right. But, you know, if you can spell it out, just try, because 10 to some power is, you know.
I don't even know the prefixes, right? Like billion, trillion, quadrillion. Like, I don't know how to go this high because I think it's going to be like 10 to the 20 something. Like,
wow.
What?
You're off by a lot.
Okay, still, yeah, okay. See, I have no way to conceptualize this.
It's 37 zeros.
Oh, okay.
50 trillion, trillion, trillion.
Oh, the triple trill.
Yeah, so not just a trillion, but a trillion trillions.
Not even a trillion
and
trillion.
Not even a trillion trillions. A trillion trillion trillions. And then 50 of those.
And multiply it by 50. Yeah, got it.
Yeah. That's 50 and 3 trillions, which is 5 with 37 zeros. And that's useful for this question because then we can figure out how long one base pair is. So there is a unit of measure for base pairs. 100 base pairs is 34 nanometers or 1 angstrom. So we could take 50 trillion 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 and make it 50 million billion billion billion, you know, basically divided by 100, and then multiply that by 34 nanometers for that 100 each. There's that many angstroms, basically, of base pairs. You end up with, just a reminder, Mike Acomo's back of the napkin was, in his original question, half the diameter of the galaxy. And in his email, when I told him we were going to have it, he said more like a fraction of the universe, which was closer, but still way off. Do you have any guesses how much it is?
Well, a fraction of the universe could be so many things. Is it greater than half the universe?
Yes, very much so, greater than half the universe.
What? Which was, again, 93 billion light years, you said? The observable universe is across? So, geez. Is it appreciably close to the full scale of the universe?
Observable universe, it exceeds. It's 1.8 trillion light years. The observable universe is only 93 billion light years. We're looking at 19 times longer than the observable universe is wide.
Yeah, I was going to say that's roughly 20. Wow.
Yeah.
Wow.
Yeah.
Just on Earth.
Just ours. Just our DNA. Just all the biomass of our planet estimated by this 2015 study. So again, 50 trillion, trillion, trillion base pairs divided by 100. So you get into angstroms, which are 34 nanometers each. So you end up with 1.8 trillion light years.
I can't believe I don't hear this more often.
Isn't that wild?
You know, I always hear the we're all made of star stuff. There's more trees on Earth than there are stars in the Milky Way. I hear those science fun facts all the time. I've never heard all the DNA on Earth is 20 times longer than the diameter of the observable universe.
That's wild.
Small side note, where did you get the 93 billion miles for the universe thing?
Observable universe? NASA.
Is it the observable universe? Because, like, we can only see 13 billion light years in any direction, right?
As of 2006, the observable universe is actually 94 billion light years across. It's a little...
It's gotten bigger.
Rounding, I guess. But good to check.
Thank you. I was just wondering how they came up with that. But if you say it's from NASA, I'll believe
it.
It's
imagine.gsfc.nasa.gov. Age and size of the universe through the years. We thought it was 300,000 light years in 1919.
And then Hubble.
I guess it depends now because it's observable, so it's probably Webb will make it bigger.
Well, again, observable, you can only look as far as light can have traveled in the age of the universe, so
right.
13.8.
Yeah, you're totally right. 13.8 billion years old. But the universe is expanding, so that inflation at some point has gone faster than the speed of light. So the universe is like 46 point something billion years in every direction that we can see. So 93, 94 billion light years across.
But no, I would have never guessed it's almost 20 times the diameter of the universe, as far as we know.
It's wild, as far as we know. Just sit with it. It's so much.
Think of what we could do if we all worked together and contributed all of our DNA strands.
Yeah.
That's a bonkers number. I can't hold it in my head.
No.
I don't know where to go from here. I don't know what to tell you after this point. I feel like we need to cue the Double Helix to the Stars 5-hour playlist and just vibe to that thought.
Hang on, Kyle. Give us some psychedelic vibe to just like sit with for a second. It's 19 times longer than the width of the observable universe. 50 trillion base pairs. I feel like I'm doing ASMR right now. Anyway, so I tried to type this into Blue Bulb Project to give us some context, and they failed. They don't have numbers this big.
Yeah, how many Golden Gate bridges is that?
They failed us. The only time ever. I feel so bad. But I also wondered, 45 billion metric tons, is that as big as a star? Like if you took all our DNA and you smushed it into a ball.
No. Well, everything's going to have a Schwarzschild radius of a certain size, right? So you could find how much you need to compress it, but it can't be more than a star. It's fairly light.
It's actually fairly light because when I Google the number 45 billion tons, I just get something from the International Energy Agency, and they're like, oh, that's how much coal we use.
Yeah.
Or whatever, like in a year.
Which used to be living material anyway.
Yeah, so it's quite small. But anyway, here's the thing I think brings us out of our psychedelic music.
Oh, okay.
DNA is so small that not only could you not see this huge strand to the stars, you wouldn't feel it if you touched it. You'd put your hand through it and it would immediately break.
Oh, no.
It has no tensile strength. It's not like a spider silk where the silk wants to stay together. So you could walk through it and it could hit you right in the face and you wouldn't even know it's there.
Oh, my gosh. Yeah. You'd break your ladder to the stars, man.
Yeah, man. Don't do it, man.
No. You gotta be careful where you're walking, man.
Yeah.
Again, it's just a number so enormous. I can't understand it. I am incapable of understanding it.
Yeah, it's impossible to wrap your brain around.
Yeah, but it would wrap around my brain many times.
A lot of times.
An unfathomable
trillion.
Many times. Plus, my brain's small.
He's just a little guy.
Just a little guy, and it's his birthday. Well, Trace, thank you for tackling that question. That was absurd.
It was absolutely an absurd question. So thanks for sending it in, Micah Como. And also thanks for sending it in, Anonymous. Thanks for sending in your question.
Well, this episode of That's Absurd, Please Elaborate, was created, written, hosted, and produced by me, Julian Hugan.
And also me, Trace Dominguez.
And produced and edited by the wonderful, wonderfully talented, and man with a fireproof mouth, Kyle Sisk.
I feel like if Kyle eats a pizza, it's just going to be like, and it'd be done.
Additional editing for this episode provided by Sam Grant.
Special thanks again to those listeners who submitted questions featured in this episode, Mike Como and Anonymous. If you want to submit a question, go down into the show notes. I teased this at the top of the episode. You can find our website, that'sabsurdshow.com, and go to that'sabsurdshow.com/ask. You can email us at hello@that'sabsurdshow.com. You can call us at 302-TAPE-SHOW, TAPESHO, and leave us a voicemail. And you could even leave us a comment on Spotify or, any number of other ways. Find a way to send us your question. Mail us a postcard. I don't know. We don't have an address, but...
Not yet, we don't. Just write Trace and Julian on an envelope. The Postal Service knows what to do with that.
They know what it's about.
If you want to vote on questions, you can do so by joining our Patreon. Thanks to all those Patreon listeners. I'm really excited to tackle the next one like we teased already. And a special shout out to our absurd administrators, Les and Adam.
Leave us a review wherever you're listening right now. The reviews really help people know that this podcast is worth their time. So if you listened to this whole episode, pick up whatever device it is that you're listening it on, unless it's the car, in which case do not pick it up. But, go to your phone or whatever and leave us a review. You can leave some words. That would be amazing. If you do that, shoot us an email. We'll send you a sticker. And if you leave a star rating, we would love it was a high one. But if it's not, let us know. Shoot us an email. Tell us what you don't like about the show. Maybe we'll fix it.
Yeah.
We'll do whatever you want.
Do whatever you please.
Leave us a review.
Please love me. I'm just a little guy. Okay. Finally, special thanks to those listening on Nebula. Nebula listeners get access to ad-free versions of episodes, and they support us and other awesome independent creators that we love and watch their stuff all the time. So if you join, you're supporting us, you're supporting them, everybody wins. To join Nebula, visit go.nebula.tv/that'sabsurdshow.
So what do we say at the end of every episode, Julian?
If you're having trouble with your air-fried pie, do not put it on that spot just under your butt cheek. That's the worst spot for it.
If your pie is too hot.
If your pie is too hot for your hands, it's too hot for your butt cheek. We give people actionable advice here on this show.
So at the end of every episode, what do we say? For those over 40, when it's hot so hot in here, you really just have to take a little flap off of your lower butt. And that's it. That'll cool you right down. See, Nelly, he had the Band-Aid on his cheek, but he should have.
Which was a sensitive area, right? But to save on the heating costs,
Thanks for listening, everyone. We'll see you next week with another episode. T.O. Blancy, Olata, got a big genome. I wish we knew why. T.O. Blancy, Olata, got a big genome. Gosh, I wish that we knew why. There's coding DNA in your cells. Go home. Da, 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 da. It's got nitrogen as bases and some phosphorus. And, it's not really junk DNA. Ha 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 ha. I just made all that up. I don't know what the hell I'm talking about.